Chapter 4 Principles of Inheritance and Variation. Question answers from Class 12, Subject Biology. Exercises. Question 1. Mention the advantages of selecting pea plant for experiment by Mendel. Answer. Pisum sativum was chosen by Mendel for the following reasons. First, the pea is an annual plant with a brief growing season. As a result, the findings can be achieved in a short amount of time. Second, pea has 7 distinct features with easily distinguishable traits such as tall and short stature, round and yellow seeds, and so on. Third, self-pollination and cross-pollination are both viable options for pea propagation. Mendel was able to create pure lines as well as carry out various crosses between plants with contrasting features as a result of this. Fourth, each generation of the pea plant produces a great number of seeds. As a result, it can provide a vast volume of data that can be statistically more reliably analyzed. For example, Question 2. Differentiate between the following. A. Dominance and recessive. Answer. Dominance. In the presence or absence of a recessive trait, a dominant factor or allele expresses itself. Recessive. A recessive trait expresses itself only in the absence of a dominant trait. Dominance example. In a pea plant, round seeds and violet flowers are dominant characters. Recessive example. In a pea plant, white flower, dwarf plant, etc. are recessive characters. B. Homozygous and heterozygous. Answer. Homozygous. For a particular trait, homozygous contains two similar alleles. Heterozygous. For a particular trait, heterozygous contains two different alleles. Homozygous. Only one type of gamete is produced. Heterozygous. It produces more than one type of gamete, two different types of gametes to be precise. For homozygous, the genotype contains either recessive or dominant, never both alleles. Example T or small t. For heterozygous, the genotype possesses both recessive and dominant alleles. Example T and small t. C. Monohybrid and dihybrid. Answer. Monohybrid. It is a cross between parents differing in only one pair of contrasting characters. Dihybrid. It is a cross between parents differing in two pairs of contrasting characters. Monohybrid example, a cross between a dwarf and a tall pea plant. Dihybrid example, a cross between a yellow wrinkled seed and a green rounded seed. Question 3. A diploid organism is heterozygous for 4 loci. How many types of gametes can be produced? Answer. A locus is a fixed point on a chromosome that is occupied by one or more genes. For an allele pair, heterozygous entities contain different alleles. Thus, a diploid entity which is heterozygous at 4 loci has 4 different contrasting characters at 4 different loci. Example, if an entity is heterozygous at 4 loci with 4 different characters such as capital M, capital N, capital O, small o, capital P, small p, then while meiotic division, they split to form 8 different gametes. Diploid organism, capital A, capital B, capital C, capital D, small a, small b, small c, small d. Capital A small a, capital B small b, capital C small c, capital D small d. 16 different gametes. If genes are not linked to each other, then the diploid entities will produce 16 different gametes. But if genes are linked, gametes will decrease their number as the genes may be linked, which in turn will be inherited together during the meiotic cell division. Question 4. Explain the law of dominance using a monohybrid cross. Answer. The law of dominance was proposed by Mendel. It states that a dominant allele expresses itself in a monohybrid cross and suppresses the expression of a recessive allele. But the recessive allele for a specific character does not vanish but remains masked or hidden in the progenies of the F1 generation, which resurfaces in the subsequent generation. Example, when a monohybrid cross between two pea plants having round seeds R and wrinkled seeds r was carried out, this one and this one, All the seeds in the F1 generation were observed to be round, capital R, small r. If the round seeds were self-fertilized, both the characters round and wrinkled seeds appeared in the F2 generation is in the 3:1 ratio. This one. Therefore, in the F1 generation, the character that is dominant, that is the round seeds, surfaced, and the recessive character, that is the wrinkled seeds, get suppressed that resurfaced in the F2 generation. So friends, this is selfing of F1 and this is F2 generation. So you can draw the diagram. Question 5. Define and design a test cross. Answer. A test cross is a cross between a plant with an unknown genotype and the recessive parent. This one and this one. To determine the genotype of an unknown plant, a test cross is utilized. That is, whether the plant is homozygous or heterozygous. If the test cross progeny has a 1:1 dominant to recessive ratio, the unknown plant is heterozygous. If the progeny are all dominant, however, then the unknown plant is homozygous. Question 6. Using a Punnett Square, work out the distribution of phenotypic features in the first filial generation after a cross between a homozygous female and a heterozygous male for a single locus. Answer. When heterozygous male guinea pigs capital W small w with black coats are crossed with homozygous female guinea pigs small w with white coats, we can see that the male generates two types of gametes, capital W and small w, while the female produces only one form of gamete, small w. Here you can see the female and the male. We can see that the genotypic and phenotypic ratios in the F1 generations are the same. That is 1:1 using the Punnett square. Question 7. When a cross is made between a tall plant with yellow seeds capital T small t capital Y small y and a tall plant with green seeds capital T small y, what proportions of phenotype in the offspring could be expected to be A. Tall and green, B. Dwarf and green? Answer. When a cross between a tall plant with yellow seeds and a tall plant with green seeds is carried out, the following is produced. 3 tall green plants, 1 dwarf green plant. Parents, tall yellow seed plant, capital T, small t, capital Y, tall green seed plant, capital T, small y, and you can see the gametes. Next, so here we get tall yellow, tall green, tall green, dwarf green, tall yellow, dwarf yellow. Phenotype tall and green plant 3. So here is tall green, 3. Then dwarf and green plant 1 here. Question 8. Two heterozygous parents are crossed. If the two loci are linked, what would be the distribution of phenotypic features in the F1 generation for a dihybrid cross? Answer. Linkage is defined as the coexistence of two or more genes in the same chromosome. If the genes are situated on the same chromosome and lie close to each other, then they are inherited together and are said to be linked genes. For example, a cross between yellow body and white eyes and wild type parent in a Drosophila will produce wild type and yellow white progenies. It is because yellow bodied and white eyed genes are linked. Therefore, they are inherited together in progenies. This is the diagram. Question 9. Briefly mention the contribution of T.H. Morgan to genetics. Answer. The contributions of T.H. Morgan in the field of genetics are as follows. He proposed and established that genes are positioned on chromosomes. He discovered the basis for variations as a result of sexual reproduction. He discovered the concept of linkage and discriminated between linked and unlinked genes. He stated the chromosomal theory of linkage. He carried out a study on sex-linked inheritance. Morgan stated a chiasma type hypothesis demonstrating that the chiasma causes crossing over. He observed that the frequency of recombination between two linked genes is directly proportional to the distance between them both. Proposed the theory of inheritance. He put forward the methodology for chromosome mapping. He carried out a study on mutation. Question 10. What is pedigree analysis? Suggest how such an analysis can be useful. Answer. A pedigree is a record of the inheritance of a specific genetic trait for 2 or more generations, which is presented in the form of a diagram or family tree. Pedigree analysis is an analysis of several generations of a family, which is used on human beings and domesticated animals. The usefulness of a pedigree analysis: it serves as a powerful tool which can be used to trace the inheritance of a particular trait, disease, or abnormality. It is helpful for genetic counselors to suggest to couples the possibility of having children with genetic abnormalities such as color blindness, hemophobia, thalassemia, sickle cell anemia, etc. The analysis is helpful in indicating the origination of a trait and its flow in ancestors. It is helpful in suggesting that Mendel's principles can be applied to human genetics with some alteration, such as quantitative inheritance, sex-related linkage, and characters. Helpful in reasoning why marriage between close relatives is harmful. Helpful in extensive research in the field of medical science. Question 11. How is sex determined in human beings? Human beings exhibit male heterogamy. In human males, XY produces two different types of gametes, X and Y. The human female XX produces only one type of gametes containing X chromosomes. The sex of the baby is determined by the type of male gamete that fuses with the female gamete. If the fertilizing sperm contains X chromosome, then the baby produced will be a girl, and if the fertilizing sperm contains Y chromosome, then the baby produced will be a boy. Hence, it is a matter of chance that determines the sex of a baby. There is an equal probability of the fertilizing sperm being a X or Y chromosome. Thus, it is the genetic makeup of the sperm that determines the sex of the baby. Question 12. A child has a blood group O. If the father has a blood group A and mother's blood group B, work out the genotypes of the parents and the possible genotypes of the other offspring. Answer: a set of three alleles Ia, Ib, and small I control the blood group characteristics in humans, where alleles Ia and Ib are equally dominant and allele small I is recessive to the other alleles. Genotypes: blood type A, Ia or Ia small I. Blood type B, IB or IB small I. Blood type AB, IB. Blood type O, small I. Analyzing the given information, the child has blood group O, genotype small I. The father has blood group A. The mother has blood group B. Determining parental genotypes. For the child to have the genotype small I, they must have inherited an small I allele from both parents. Therefore, the father's genotype must be Ia small I heterozygous A. The mother's genotype must be Ib small I heterozygous B. Possible offspring genotypes. To find the possible genotypes of other offspring, we can use a Punnett square. Ia, Ib, Ia small I, small Ib small I. From the Punnett square, the possible offspring genotypes are IAIB blood type AB, IB small I blood type B, IA small I blood type A, small I blood type O. Therefore, the possible blood types of other offspring are A, B, AB and O. Question 13. Explain the following terms with examples. A. Codominance. Answer: codominance occurs when both opposing alleles of a gene pair are equally expressed. For example, in the human blood group, both A and B are equal prominent, and when both are present, the AB blood group appears instead of either A or B. B. Incomplete dominance. Answer: incomplete dominance occurs when a dominant allele does not entirely cover the effects of a recessive allele, resulting in a blending of both alleles in the organism's physical appearance. It's also termed as half dominance or semi-dominance. Take, for example, the flower color of a snapdragon plant. The red flower color R has an imperfect dominance over the white flower color r. When both alleles R are present in the same plant, capital R is unable to completely repress small r, resulting in a pink flower phenotype. Question 14. What is point mutation? Give one example. Answer. Point mutation is a change in a single base pair of DNA by substitution, deletion, or insertion of a single nitrogenous base. An example of point mutation is sickle cell anemia. It involves mutation in a single base pair in the beta globin chain of hemoglobin pigment of the blood. Glutamic acid in short arm of chromosome 2 gets replaced with valine at the sixth position. Question 15. Who proposed the chromosomal theory of inheritance? Answer. The chromosomal hypothesis of inheritance was presented by Sutton and Boveri in 1902. This states that chromosome serves as carriers for gene inheritance. Question 16. Mention any two autosomal genetic disorders with their symptoms. Answer. First, sickle cell anemia. When glutamic acid is replaced by valine in the sixth position of the beta globin chain of hemoglobin due to a point mutation, the biconcave shape of hemoglobin transforms into a sickle shape. This causes a reduction in blood oxygen delivery as well as weakness and fatigue. Second, phenylketonuria. Phenylketonuria is a condition in which a person lacks an enzyme that transforms amino acids and phenylalanine to tyrosine. The phenylalanine then builds up and is converted to phenylpyruvic acid and other derivatives. Mental retardation is a symptom of this disease. They are eliminated in the urine due to inadequate renal absorption. Friends, we have completed all these question answers from chapter 4, Principles of Inheritance and Variation. If you like the video, kindly like it and share it with your friends. You can support me by joining the channel membership for donations and early access to new videos. We will meet in the next video. Thank you for watching.